So our topic for today is about gene expression from transcription to translation. Ayan. So we're going to talk about how this transcription happens or how does a translation happens in a gene, ayan, in a DNA sequence. Ayan. So what is the learning objectives for this lesson? So at the end of the lesson, the student should be able to explain the concept of gene expression and its importance in living cells. B. Describe the process of transcription, including key participants, which is RNA, polymerase, nucleotides, and mRNA. So, and last but not the least, identify the location of translation in the cell. Yeah. So, first, we're going to talk about what is the relationship between genes and proteins. So, genes has the capabilities to create proteins. And so, there are three actually dito sa there are three researchers who performed studies about the relationship between genes and proteins. So the first scientist who performed the study is Garrod in 1908, where he proposed that genes control metabolism by affecting enzymes. Enzymes is made out of protein. And so makikita natin yung relationship between genes and enzymes or proteins, kung saan genes control metabolism by affecting enzymes. Enzymes, ang trabaho ni enzymes is to help the metabolism process inside our digestive system. So the genes control it by affecting our enzymes. However, in Garrod's situation, hindi masyadong pinagtuunan ng pansin because yung mechanism niya was unclear para sa mga tao noon. So, inignore nila yung proposal ni Garrod. However, na-perform ulit ang studies na relationship between genes and proteins nung kila Biddle and Tatum noong 1940s, where they showed a direct link between genes and specific enzyme. So, one gene is to one enzyme. So, in 1956, ayan, si Ingram linked mutations in a single gene to change a single amino acid. Amino acid is the synthesis of proteins. Kaya, during the mutation, gene mutation, may nangyari doon which change the amino acids, which resulted to a sickle cell anemia. So, dito, makikita natin yung relationship between genes and proteins. Kasi whatever is happening on genes, it affects the proteins as well. When genes can control proteins. And so, they are interconnectedness to one another. Ayan, so, the next is the transcription. So, what is transcription nga? So, transcription is how a cell copies the instructions from DNA into a special molecule called mRNA or messenger RNA. So, ang messenger RNA na to, it carries the information to build proteins. So, may mga enzymes na tumutulong dito which called RNA polymerase or DNA-dependent RNA polymerase. They're responsible for the copying using the DNA strands as a template. Yan. So, template nila is yung DNA strands para makapag-build or makapag-copy ng complementary RNA strand. And so, that's how the transcription happens. So, we're going to talk about how does transcription happens in prokaryotic cell. Yan. So, first, we're going to identify what is prokaryotic. Yan. So, prokaryotic, it is a unicellular or a single-celled organism, which the famous term for prokaryotic cell is it lacks nucleus. Compared to eukaryotic cells, may nucleus siya. Prokaryotic does not have nucleus and no membrane-bound organelles. So, prokaryotic cells, ang mga under dito is bacteria, such as E. coli, ayan. So, naka-under sila sa prokaryotic cell. So, in prokaryotic transcription, ito yung process in which mRNA transcribes the genetic material in prokaryotes are produced. So, or to be translated into proteins. Ayan. So, Katulad nga sabi ko kanina, kukuha sila sa, ang gagamitin ng template is yung DNA strands in order to create a copy of itself para makapag-produce ng proteins. So, may 3 steps sa prokaryotic transcription, which is initiation, elongation, and termination. Ayan. So, pag-aaralan natin yung tatlong steps na yan. So, sa 3 steps na to, first, we're going to talk about initiation process. So, in the initiation process, the RNA polymerase holoenzyme, which consists of RNA polymerase, and the sigma factor. So, dito sa RNA polymerase kasi na to, meron siyang 5 units. First is the beta factor. Beta, dalawang alpha at isang omega. And so, ang pinakamalakas sa kanila each is si beta factor. Tapos, ang pinakamaliit ay si omega. Yan. So, lima silang units ng RNA polymerase. The next is the sigma factor. So, si sigma factor, kung wala siya, walang initiation na mangyayari. Kasi si sigma factor ang tumutulong sa RNA polymerase para makita niya yung promoter. In order for the promoter to bind the region of the DNA. Ayan. So, the next process is, or the next step is elongation. So, during the elongation process, umalis na si sigma factor dito. So, the RNA polymerase moves along the DNA strands, adding nucleotides to the new RNA strands in a 5-3 direction. Dito na nangyayari yung copying nung RNA polymerase doon sa DNA strand. Ayan. So sa termination naman, dito nahihiwalay yung na-copied na from the DNA. Ayan. So, from the template, alis na siya doon. So, may 2 types ng termination sequence, which is RHO-dependent and RHO-independent. Yan. So, tatapik natin yan. So, the RHO-dependent, it is a termination sequence that requires protein. Napaka-dependable ng RHO na to sa protein na yun. Ang tawag sa protein na yun is RHO factor. In order to release the RNA polymerase sa DNA. Ayan. So, our RHO-dependent termination requires ATP. Ayan, ATP to ADP in order na makaalis ito doon sa DNA strand na yon. Sa RHO-dependent. While sa RHO-independent naman, ayan, from the word itself, hindi niya na kailangan ng protein. Hindi na kailangan ng protein dito. Kasi, independent na siya. Kaya niya nang i-handle ang sarili niya. Ayan. So, during this sequence, magpo-form siya ng hairpin loop in the RNA strands which stalls the RNA polymerase and it causes the fall of the DNA. So, dito sa first picture, makita natin yung inverted repeats ng DNA. To transcription, during transcription process, magpo-form siya ng hairpin structures kasi magkoconnect sila guanine cytosine, ayan, uracil to uracil, ayan, magkakakonect in order to form a hairpin-like structure. So, sa second picture naman, ayan, so magpo-fall off na itong structure na to because RNA is released ending the transcription. Kasi itong adenosine, ayan, yung U base pairs niya kasi are the weakest. So, bibitaw na siya doon sa strand na yon. Ayan. So, doon naman tayo sa transcription ng eukaryotic cell. So, takes to Sir Robert Roder, where he discovered in 1969 that eukaryotic cells have 3 distinct transcribing enzymes. So, ang tawag dito is RNA polymerase 1, 2, and 3. Mayroong 4 at 5 ng RNA polymerase which are really not essential for life, pero makikita ito sa plants, meron silang 4 at 5. So, transcription factors. So, these proteins play a vital role virtually every aspect of transcription from transcription process. from binding of polymerization the DNA template to initiation of transcription to its elongation and termination. So, same process lang sila ni prokaryotic cell. Meron silang initiation, elongation, and termination. However, mas complicated kasi sa eukaryotic cell compared sa prokaryotic cell. Ayan. So, may transcription factors na tumutulong na tumutulong kila RNA polymerase kasi hindi kasi nila makita or hindi nila ma-detect si promoter. So, they need transcription factors. Parang kanina si prokaryotic cell, in order to transcribe sa DNA, kailangan niya si sigma factor. Dito naman, kailangan din ni eukaryotic cells ng mga transcription factors para ma-detect nila si promoter, para mag-start din ang initiation process. Ayan. So, may primary transcripts tayo, which are typically have fleeting existence being processed into smaller functional RNAs by a series of cut and paste reaction. Ayan. So, meron nito yung transcription units. Ayan. Where ang primarily function nila is to help RNA polymerase in order to mitigate the transcription process. Ayan. So, RNA, it is a molecule or a single helix bond na made out of nucleotides which are ribosugar attached to a nitrogenous base and a phosphate group. Ulitin natin. Ribosugar attached to a nitrogenous base, tapos phosphate group. Ayan. So, meron siyang 4 na nitrogenous base which is adenine, guanine, uracil, and cytosine. Ayan. So, meron 3 types ng RNA which is messenger RNA. So, si messenger RNA, ang responsibility niya is to delivers the copy of DNA to a ribosome. So, mRNA kasi, it transcribe from DNA and contains a genetic blueprint to make proteins. Ayan. So, i-deliver niya na yun sa ribosome. Yun ang copy niya na from the DNA strand. Ayan. So, transfer RNA naman, they are responsible to translate mRNA into proteins. Ayan. While ribosomal RNA, it forms ribosome. They are responsible to create ribosomes which are essential for protein synthesis. Ano ang byproduct nito? Transcription process. Transcription to translation to form a protein. Ayan. So, ang byproduct nito ay protein.
Let's proceed to synthesis and processing of mRNA. Transcription is a process by which DNA gets converted into mRNA, also known as messenger RNA. We all know that DNA is a double strand, while RNA is a single. The synthesis of mRNA from DNA occurs in several stages. First, initiation. Second, elongation. Third, termination. So, initiation. It occurs when the enzyme RNA polymerase binds to a region of gene called promoter. So, nakita nyo lang sa picture na yung dito sa may tinuturo ng dito sa may red, sa may baba. Makikita nyo dyan yung promoter region. And, dito mag-start yung RNA polymerase dito sa nabinugan na green. So, during initiation, RNA polymerase binds to a promoter region of DNA. The promoter region is a short sequence of DNA that signals the start of a gene. RNA polymerase then causes the 2 DNA strands to separate and begins to add nucleotides to the growing mRNA strand. The RNA polymerase synthesizes mRNA starting from the 5' end going to the 3' end. So next, elongation. A regulated process in which an RNA chain complementary to the template strand of DNA is synthesized as RNA polymerase move along DNA. So, elongation is the next step of transcription. In this step, RNA polymerase continues to add nucleotides to the mRNA strand until it reaches the termination sequence. The termination sequence is a specific sequence of DNA that signals the end of a gene. Once RNA polymerase reaches the termination sequence, it releases mRNA strand and detach from the DNA. So, lastly, termination. Termination, the final step of a gene expression which plays an important role in making an end of RNA without affecting unnecessary gene expression from downstream genes. So, the last step of transcription is termination. A poly A tail is added to the 3 end of a mRNA strand. Kung makikita nyo dito sa may dulo, dito sa may 3' end, dito malalagay yung poly A tail. Dito. So, the poly A tail is a non-coding sequence of nucleotides that helps to protect the mRNA strand from degradation. Let's proceed to the next, which is small regulatory RNAs and RNA silencing pathway. Small regulatory RNAs are a class of non-coding RNA molecules that play a crucial role in regulating gene expression in eukaryotes. These molecules are typically 20 to 30 nucleotides in length and bind to a complementary sequence of messenger RNA, or also known as mRNA, thereby silencing its expression of a target gene. So, there are two main types of sRNAs. Small interfering RNAs, or also known as siRNAs, and microRNAs, also known as miRNAs. First, siRNAs are double-stranded RNA molecules that are typically derived from exogenous sources such as viruses or transposons. Second, miRNAs are endogenous RNA molecules that are transcribed from genes in the genome. So, dito tayo, makikita nyo dito yung siRNAs and miRNAs. So, dito tayo mag-start sa small interfering RNAs. So, small interfering RNAs, dito ay they process by the enzyme dicer into short duplexes that are incorporated into a protein complex called the RNA into silencing complex, or RISC. The RISC complex unwinds the siRNA duplex, and the single-strand siRNA molecules guides the RISC complex to complementary RNA targets. Once bound to a target mRNA, the RISC complex cleaves the mRNA, effectively silencing gene expression. So, dito tayo sa microRNAs. So, dito naman, they are folded into hairpin structures and are processed by a series of enzymes, including DROSA and lyser, into mature miRNAs that are single-stranded like siRNAs, mRNAs, or microRNAs are incorporated to RISC complexes. However, instead of cleaving targets mRNAs, microRNA typically binds to the 3' untranslated region, or UTR, or target mRNAs and repress translation. So, next, RNA silencing pathways are a collection of cellular processes that are used small regulatory RNAs to regulate gene expression. These pathways are essential for normal development, differentiation and function of organisms. They also play a role in defense against viruses and transposons. So, RNA silencing pathways are essential to the body toolbox for using tiny RNA molecules to control gene activity. These pathways are crucial for an organism to develop normally from different cell types and function properly. They are also act as a defense system against invaders like viruses and transposons or jumping genes. Next, there are several steps involved in RNA silencing pathway. First, production of small regulatory RNAs. Second, processing of small regulatory RNAs. Third, RISC complex formation. Fourth, target recognition. And last, gene silencing. So, first, siRNAs can be derived from exogenous double-stranded RNA or from long double-stranded RNA molecules that are produced endogenously. miRNAs are transcribed from gene in the genome and are folded into hairpin structure. Second, siRNAs and miRNAs are processed by enzymes such as dicer into mature single-stranded RNA molecules. Third, mature siRNAs and microRNA are incorporated into RISC complexes. Fourth, the single-stranded siRNA or microRNA molecules in the RISC complex guides the complex to complementary mRNA targets. And last, once bound to the target mRNA, the RISC complex can either cleave the mRNA or repress translation or the microRNA. So, the specific outcome of RNA silencing depends on the type of sRNA and the nature of its target mRNA. MicroRNAs typically cause translational repression of their target's mRNAs, while small interfering RNAs often lead to mRNA degradation. RNA silencing is a powerful tool for studying gene function and has the potential to be used to therapeutic purposes. For example, researchers are developing synthetic small interfering RNAs that can be used to silence the expression of disease-causing genes. So, next reporter.
So let's now proceed to the encoding of genetic information. When we say genetic information, it is recorded in a DNA as a nucleotide sequence, which is relevant for constructing proteins, which are also necessary for the functioning of cells and the whole organism. Proteins are constructed of amino acids. So we are all familiar that the building blocks of proteins are amino acids. Did you know that there exists various number of amino acids? But nature or commonly uses only 20 quite specific amino acids. There are 20 amino acids and 4 bases. To sum up, the total number of 3-letter combinations of element is 4 to the third power, which is equal to 64. So let's now proceed to the properties of genetic code. The first to put forward the idea of a genetic code was a well-known American physicist named George Gamow. Genetic code is a triplet. Each letter of the genetic code consists of a sequence of 3 nucleotides. 3 nucleotides forms a codon, and 1 codon encodes 1 amino acid. So for example, the nucleotides GCU, or the guanine, cytosine, and uracil. If you combine these 3 nucleotides it forms a codon and read as an alanine, which is an amino acid. So the overall genetic information is stored in the chromosomes of an organism is called the genome. So let's now proceed to the next slide. So as you can see in the next slide, that is the genetic code wheel. The genetic code used in the image are RNA nucleotides. So as you can see in the central circle, thymine is replaced by uracil. This is just like what I said a while ago, that is the genetic code wheel. The first letter of a codon is located in the central circle. Next ring is the second letter, and then comes the third. On the outer side of the circle are the names of amino acids. So let's use this genetic code wheel. So for example, codons UCA. So let's first locate the uracil, which is located in, the first letter is located in the central circle. It is located on the upper part, right upper part of the circle. And the second letter is C. C is located at the second ring of the circle. And then the third letter is A, which is located in the third ring of the circle. So as you can see, UCA is under the amino acid named serine. So ganun lang gamitin yung genetic code wheel. So I have a question here. Did you know that there are few meaningless codons that do not match any of the amino acids? Yes, there exist 3 meaningless codons. However, they might be meaningless but not useless because such codons have a special function. They serve as stop signs. So, yung mga codons na ito are UGA, UAA, and UGA. These are the stop codons where termination happens. So let's now proceed to the next slide, which is the decoding the codons, the role of transfer RNAs. Transferring information from DNA to protein is called translation. mRNA carries the information from DNA. So, decoding the information in a mRNA is accomplished by transfer RNAs, which act as adapters. This transfer ribonucleic acid helps decode a mRNA or messenger RNA sequence to become a protein sequence. It also serves, this transfer RNA serves as a physical link between the mRNA and the amino acid sequence of the protein. So to further understand the tRNA, let's understand the basic structure of tRNA molecule or the transfer RNA molecule. So as you can see in the next slide, these are the parts of tRNA. So the first one is the tRNA molecule has 5' terminal phosphate group. So as you can see in the image, it is located in the upper left corner. The second part is the acceptor stem or the acceptor arm. It has a 7 to 9 base pair stem and the 3' terminal nucleotide which contain the codes CCA. That code is located in the upper right corner, which is used to attach the amino acids. Next part is the D-arm loop or the D loop. It is a 4 to 6 base pair stem ending in a loop that often contains an unusual molecule called the dihydroridine. The third part is the anticodon loop. The anticodon loop of the tRNA is a 5 base pair stem and its loop called the anticodon. Anticodon, which are 3 nucleotides that recognizes the mRNA or messenger RNA codon for its amino acid. The fourth part is the T-Psi-C loop, where psi is from pseudoridine, from the word pseudoridine. And lastly is the variable loop, which helps identify the specific tRNA molecule, where its size varies. The Wobble hypothesis by Francis Crick. So the Hubble hypothesis explains the possible degeneracy of codons since there are one more than codon for one amino acid. So, according sa hypothesis na ito, tanging ang dalawang codon sa unahan ang may precise na kapares sa bases ng anticodon ng transfer RNA. Kung saan naman yung third or yung pangatlong pairing sa codon at anticodon ay nagtataglay ng Hubble characteristic. So when we say Hubble, it means sway or move unsteadily. That is why Crick come up to this for hypothesis or conclusion. So the first hypothesis that he come up is the first two bases of the codon make normal or canonical H-band pairs with the second and third bases of the anticodon. At the remaining position, less stringent rules apply and non-canonical pairing may occur. The Hubble hypothesis thus proposes a more flexible set of base pairing rules at the third position of the codon. The third hypothesis, the relaxed base pairing requirement or Hubble, allows the anticodon of a single form of transfer RNA to pair with more than one triplet in messenger RNA. The last one hypothesis. The rules first base U can recognize A or G. First base G can recognize U or C. And first base I can recognize U, C, or A. So why does the Hubble hypothesis important to the genetic code? It is because Hubble pairs are fundamental in RNA secondary structure and are critical for the proper translation of the genetic code. In addition, the existence of Hubble minimizes the misreading of a codon of a code. So for example, the amino acid leucine, it has a codon CUU, where we're also misread as CUC or CUA or CUG. During the transcription of messenger RNA, the codon would still be translated as leucine during protein synthesis. So you may refer to our genetic code wheel to verify if it is correct. It is in the previous slide. So let's now move on the the tRNA charging or amino acid activation. So move our discussion to the tRNA charging or amino acid activation. So when we say transfer RNA charging, it is the process of attaching an amino acid to its corresponding transfer RNA, which is also called as amino acid activation, also known as tRNA charging or amino acylation. So in this process, we only have two major steps. But before we proceed to its process, let us know first the mechanism of transfer RNA charging. It has amino acid binding site. It has also adenosine triphosphate binding site and the transfer RNA binding site. So in this process, we catalyze and use an enzyme, amino acyl tRNA synthetase. So let's now proceed to the major step. The first step is amino acid binds to adenosine triphosphate, forming amino acid AMP, releasing pyrophosphate or 2-phosphate. So these 2-phosphate are loose during the process of phosphorylation. So now, amino acid is activated after the phosphorylation process. So next to that process is the formation of charged tRNA, where amino acyl AMP enzyme binds to tRNA, forming amino acid plus tRNA. Okay, let's now see the reaction. The uncharged tRNA binds to the active site of the enzyme. So this enzyme transfer amino acid. So from amino acyl AMP to tRNA to form amino acyl tRNA. So in the next step, AMP is released. The charged tRNA is also released. So that's the process of amino acid activation. So, this process of joining tRNA with its corresponding amino acid is called the tRNA charging or amino acid activation. Thus, the stored energy is used in the formation of peptide bond during translation. So let's now proceed to the translation process since um, we formed we already formed the peptide bond in the amino acid activation. So first, let's define what is translation. So translation is the process by which genetic information encoded in DNA is converted into functional proteins. So in this process, there are three key players, which are the mRNA, ribosomes, and tRNA. So mRNA carries the genetic code copied from DNA during transcription. On the other hand, ribosomes are large complexes of RNA and protein that act as the protein synthesis factories. And the last one is the tRNA, which is the adapter molecules that carry a specific amino acid. So, let's now start the process of translation. The step 1 is initiation. It recognizes and initiates protein building, and the translation begins in the cell's cytoplasm. Take note na nag-i-start yung translation process in the cell's cytoplasm. When the subunits of a ribosome recognize and assemble around a codon on the mRNA transcript called the start sequence. So, the starting codon is AUG, decoded as methionine, in which it is always the first amino acid in any protein that is produced. So the step 2 is the elongation. After the mRNA start sequence, which is the codon AUG, the next three bases of the mRNA specify which tRNA molecule should bind to the mRNA. So tRNA molecules wherein each are carrying a specific amino acid binds to the complementary codons on the tRNA. So this process of progressively linking together the amino acids is specified an mRNA strand is called the protein synthesis. The last step is the termination. Eventually, the ribosome arrives at the codon on the mRNA that signals the end of translation. Once the ribosome encounters this stop sequence, the assembly of the amino acid chain is complete and the amino acid chain and mRNA molecule are released from the ribosome. Remember, our stop codons are UAA, UAG, and UGA. This is where the termination occurs. So, that's all for our report. Thank you for listening. I hope you learned something today.